And next we have saving slimy salamanders. Try saying that fast, a few times. Using modern techniques on legacy collections. Kaylin Meyer.
Hello. This talk is also by Amanda Lawrence, but it's 5 minutes and only one of us can speak, so I either won or lost that coin toss, depending how you feel about public speaking. Oh, there he is. All right, so herpetologist Richard Heighton began collecting salamanders along the Appalachian Mountain Train in 1957, and he continued collecting them until the early 2000s. His collection is known as the Heighton Collection to us, and it offers an exemplary case study of change over time across the entire mountain chain. And he went from New York down to Florida and as far west as Arkansas. We managed to acquire the collection in 2012, and it included several thousand frozen, unprocessed specimens. So in 2017, Amanda Lawrence and I started a year-long dissection and processing project on them. And this is their story, if you would. So over a period of roughly 8 months, we dissected and prepped about 2,500 of those salamanders. The field tags and the notes that accompanied each specimen were transcribed into a series of notebooks and also into an electronic database. And each specimen is associated with multiple various numbers. They each have a field number that was assigned by Heighton. They have a bleeding number that is associated with Heighton's own research. So while they were unprocessed, Heighton did go ahead and bleed them for early DNA studies. And there's also our own specimen number for them, and there is a biorepository tissue sample number. So there's a whole lot of various research informatics going on with this project as well. And from the beginning, we took a holistic sampling approach to processing this collection. So the preparation process involved swabbing their bellies, their limbs, and their tail to capture each specimen's microbiomes. And in particular, we were interested in knowing if there was any evidence of the chytrid disease in these early populations. For if you don't know, chytrid is an infectious fungal disease that is impacting all amphibians. It's spreading worldwide, and that's as a result of global trade and climate change, as well as a couple of other factors. And it can devastate entire populations of amphibians and especially salamanders. We also took tissue samples from the liver, and those were then frozen in nitrogen and stored in our biorepository. And then the salamanders were properly positioned, and we fixed them in 10% formalin, then slowly transferred them over to ethanol. And the journey for an individual salamander took roughly 6 to 7 whole business days. Well, we learned several things from this, what we called a pilot program, because we never undertook anything of this size before. First, a dedicated team of 2 is pretty much the ideal size. A single person was really easily experiencing fatigue and burnout due to the extreme time constraints and the workload of the project. And more than 2 dedicated staff resulted in one of those like too many cooks in the kitchen sort of scenarios where we were possibly repeating or skipping specimen numbers, so that was just right out. We also learned that not all the salamanders are equal, and this kind of is one of those things that's really obvious, but you don't think about it until you actually encounter it. There was significant size difference between these specimens. So that's actually a specimen that's about 4 inches long, if my memory serves me correctly. Some were longer than 5 inches, and some were as small as a quarter. Think about that, a quarter. Finding a liver on something the size of a quarter. It's not fun. They took significantly longer to dissect, and they were extremely difficult. The larger specimens, on the other hand, took significantly longer to thaw out before we could actually begin processing them. And there's one species in particular known as Plethodon glutinosus that were definitely the worst species we had to work with. For non-herps in the room, this is a species of salamander that basically exudes a super slimy glue when they're threatened. And it's easy to say that these salamanders definitely died angry, and their slime is probably more gross from being frozen and then thawed. There was no recovering those gloves. We changed gloves after every single specimen. It was horrible. And importantly, the ability to swab and test each specimen's microbiome was an amazing idea and an amazing opportunity, and I'm glad that we thought of doing it, because we have a researcher now studying the swabs for evidence of that chytrid, and she has found a handful of them with that disease already. And so that suggests that chytrid was possibly in the area much earlier than we previously thought, so I'm really looking forward to seeing the results of that research. And overall, that idea of taking specimens as a holistic example of their whole environment, I think, is a really important idea for natural history museums to look about and think about moving forward. Like, it is not just a single specimen. That specimen came from a whole environment, and it would be good if we started thinking of it as something that we should sample everything of and capture as much data as possible within that environment. I'm about out of time. But if you'd like to hear more about this project, Amanda and I are presenting a full-length talk on it, starting at 3:30 in Ballroom B. So hopefully if you're more interested, you can learn more then.
I just want to mention if Jim Holstein is still in the room,